Okay, so I know you've already got the videos for the immune system chapter, and they're very extensive, but I want to make a review video for those who are struggling to try to fit all of the pieces of the adaptive immune system together. So in this video, I'll try to do some more dynamic drawings as if I had a whiteboard, and we'll talk about how the humoral immune system works. So we'll talk about phagocytizing a pathogen, activation of the B cell, the role of the helper T cell, so on and so forth. I think for the cellular immune system, the lectures from last week are enough, so I'm not going to go over that piece again. You can just go back to that link in last week's learning module. But sometimes having a more dynamic view, if I had had you face to face, we would be using the entire whiteboard to try to piece all of this together. So in this drawing, you have a naive B cell. So this is a B cell. It makes immunoglobulins. So if you look here, this is the immunoglobulins that I've drawn. Immunoglobulins are B cell receptors and antibodies. They're both the same molecule. This is a B cell receptor immunoglobulin because it's still attached to the B cell surface. If the B cell were to spit these out into the body fluids, then they would be little disconnected weapons and we'd call them antibodies. They're still the same molecule. It's just a B cell receptor is attached to the B cell surface. Antibody is free floating. So here in red, you have the immunoglobulins. Here in black, you have the MHC class 2s. So when remember the B cell is a phagocyte, and it's not just a phagocyte, it's an antigen presenting cell. That means it's a phagocyte that after it phagocytizes a pathogen, it presents remnants of what it digested on the MHC class 2s, and it also has MHC class 1s, like all of your cells. So MHC class 1s, that's the self-tag. So we're going to not really work with MHC class 1s, but I'm putting these all up here because I think one of the things that gets confusing is that a cell has more than one receptor on its surface, right? I mean, there's many more receptors than just these. These are just the ones that we're focusing on. So I want you to remember that a B cell has B cell receptors, it has MHC class 2s, and it has MHC class 1s. All cells have MHC class 1s, only B cells have B cell receptors, and only antigen-presenting cells, which include B cells, dendrocytes, and macrophages, which we haven't covered yet. We've just covered the B cell, but all of those three antigen-presenting cells have MHC class 2. So all cells in green, just B cells in red, and all antigen-presenting cells in black. Okay. So let's take a look at how B cells become activated. And so I'm going to keep cutting out to make these shapes instead of drawing them while you're watching for the most part because it's taking me a long time since I'm just getting used to this program. So what I've done is I've copied and created a couple other naive B cells because I want to demonstrate how every B cell receptor is different. They all have different shapes, and so they're not all going to be able to bind to the same pathogen. Now remember, when these B cells are naive, every time they divide to make a daughter B cell, you get a mixing up of the genes that code for this B cell receptor. So every time this B cell divides, let's just say this original one is the mother B cell and it made these two offspring. See how its offspring don't have the same B cell receptor as the mother, and they have different B cell receptors from each other. And that has to do, again, with a high mutation rate in the genes that code for the B cell receptor, and also with mitotic genetic recombination in the genes that code for the B cell receptor. Remember, T cell receptors are the same idea. So the implications of this are that not all of them are going to be able to bind to a pathogen that enters your body. You're hoping when a pathogen enters your body for the first time, you're just hoping that you happen to have a B cell that has a receptor capable of binding to some antigen on the surface of that pathogen. So let's take a look at what that would look like here. So let's just say we have this pathogen here, and if I were doing this on the whiteboard, it would be so much more clear, but I'm just going to do my best. So there's some antigens. It has antigens of this shape on the surface of this pathogen. And I'm trying to draw these antigens on the surface of this pathogen so that it looks like they match with these B cell receptors right here. And I know that it's imperfect, but that's what I'm attempting to do. So these are going to be found across the surface of this pathogen, and I'll just write in the center here. This always shows up as 60 font when it should be smaller. Okay, so this is the pathogen. There you go. Okay, so now this pathogen is, this B cell receptor is capable of binding to this pathogen. I also want you to remember that, again, all cells are complex, pathogenic cells are complex too. There's always going to be more than one antigen to bind to, which means a single pathogen might end up activating more than one B cell, but we're just going to focus on one B cell here. Any B cell that's capable of binding to something on the surface of the pathogen could be activated by that pathogen. So you might have multiple pathways working in synchrony to fight this pathogen in the end. So if I were to then, let's see, I wonder if I can move this whole thing. All right, so if I were, oops. So this B cell and this pathogen will eventually come in contact with each other. And I can't move it. Oh, does this move? Holy moly. I can't move this from, there we go, okay. I'm sorry for the technical difficulties. So this pathogen and this B cell receptor will eventually line up together. And that is going to, so when these B cell receptors bind to the antigens on this pathogen, it's going to stimulate phagocytosis, which I will now cover in a clear sheet of paper. So remember now that once the pathogen binds to receptors on the B cell surface, the cell membrane is going to invaginate until making sort of a pocket until that pocket pinches off and makes a food vacuole around the pathogen. And then if you review back, you may remember that then you get all these little food lysosomes. So these are going to be lysosomes. Lysosomes are the digestive organelles. So they're full of digestive enzymes and acids that are going to digest this pathogen. So now this pathogen is going to be put into little tiny pieces. I'm not sure. Whoa. Well, I don't have an eraser, but this pathogen is going to be digested into tiny, tiny pieces. I sort of thought this was going to be an eraser, but I'm not sure what it is. So with that in mind, that digestion part being over, that is when we're going to start using the MHC class 2s. So I just moved the cell back up to the top left so we have more space. So that pathogen was phagocytized. It's been digested in the phagolysosome. So there's all the fragments. Most of those fragments are going to be exocytosed, so spat out into the extracellular fluid. But some of them are going to be presented here on the MHC class 2s. So this has all been B cells. B cells are phagocytes. Their first job is to bind to a pathogen, endocytose it, digest it, spit out the debris, but then its next job is to become activated, because remember, B cells have this capacity to form cellular memory of previously encountered pathogens. That's the immune system. That's generating immunity. But that hasn't happened yet, because for that to what that means is generating immunity means generating an army of long-lived B cells that will be there to fight that pathogen when it returns. Right now, you've just had this happen once. This pathogen is replicating in your body at a rapid pace. Its numbers are growing. One B cell is not enough to fight them. It needs to make more. So one of them has been digested, but the B cell activity has yet to be activated. That's what these MHC class 2s are going to be for. So I'm going to delete the label here. Just remember they're MHC class 2s. Because I'm going to delete it. Okay. So now, what talks to the MHC class 2s? Well, it's helper T cells. We don't want to kill this B cell because it just did a good job. it just killed this pathogen. So we don't want to talk to cytotoxic lymphocytes, which come and kill your cells. No, we want to talk to the helper T cell, the T cell that's going to help activate it and help with its immune response. So let me make that magically appear in the next slide. So magically, a helper T cell has appeared. They're also abbreviated TH cells for helper T cells. So that's in blue here. The helper T cells have MHC class 1s in green, just like the B cells. We are not using them right now. We're not using them because this is just, I'm just emphasizing that all cells have MHC class 1s. It doesn't matter what it is. It doesn't matter if it's an immune cell, an epithelial cell. Those are the self tags. So this is the self tag to tell the natural killer cells, don't kill me, I belong here. And also, if this helper T cell were to become infected, then it could indicate that it was infected by displaying antigens here. But it's not infected, so we're not using them right now. But they are there. All T cells have a T cell receptor, which I've indicated in blue, T cell receptor, TCR. The T cell receptors are very specific to unique pathogens, just like the B cell receptors are. And so another word for the helper T cell is a CD4 plus T cell. The reason they're called CD4 plus is because they have these, in orange I've put a cluster of differentiation number 4. These are just clusters of molecules, okay? It's a cluster of differentiation, meaning differentiate is, how you tell one thing from another. You're differentiating between them if you're discriminating between one and another. So this is one way to identify the T cells, one T cell from another, but also they have jobs. So the job for the cluster of differentiation number 4 is to dock with the MHC class 2s. Sorry, we've lost the label here. Let me put it back in black. Change the font to 20. MHC2. That's what this one was, right? And actually, I think we had this at 18. So in black, we have the MHC class 2s. The job of the MHC class 2s is to display these antigens on the B cell. And so the cluster of differentiation 4 binds to the MHC class 2s. The T cell receptor is going to find this antigen on the MHC class 2s. So if this T cell receptor is the right shape for this antigen, remember every T cell has a different T cell receptor, unique to an individual antigen. So these two cells are only going to activate each other if this T cell receptor has the correct shape to bind to this antigen that this B cell is displaying. So just to recap, the B cell bound to a pathogen because the B cell receptors matched that pathogen. The pathogen was phagocytized, digested. Antigens are presented on the MHC class 2 for the helper T cell. The helper T cell can only become activated and can only activate the B cell if the T cell receptors match this antigen. So let me see if I can move this closer here so we can see this happening. Let's see, I think we can do this faster. There we go. All right, so we have successful binding. Now, both these cells are going to become activated. The B cell is going to proliferate rapidly, and every daughter cell is now going to be a true clone. Every single daughter cell that's produced is going to have the exact same immunoglobulin that bound to this pathogen in the first place. So remember, there were all these different B cells at first, and every daughter cell had a unique B cell receptor. That's not going to happen anymore. Now this is no longer naive. So let's change that. Ooh, whoa. This is no longer a naive B cell. Oops. I don't know if I can change it. This is really bulky. I apologize. I tried to practice a little bit before starting this, but holy crap. Well, maybe I will just delete the whole thing and write it again. So now this is not a naive B cell. This is a B cell that has become activated. It's successfully encountered a pathogen. The T cell has co-activated it, so that checks and balances system has worked. And so this is no longer going to make the genetic recombination for the B cell receptors is going to stop. Mutation for the B cell receptors is going to stop, and we get exact clones over and over and over again of this exact B cell with these exact B cell receptors that bound the pathogen in the first place. This T cell receptor is also going to become activated, and it is going to proliferate rapidly. And it's going to make a lot of daughter cells with this exact same T cell receptor that bound to this antigen here. So one point I want to make is that the antigen that started this process is not necessarily the same antigen that created activation. This B cell receptor and this T cell receptor do not necessarily match. They both bind to something on the pathogen, but they might have bound to different things on the pathogen, because remember, this B cell receptor bound to an antigen on the surface, then it digested the pathogen, and any bit of debris can be displayed up here. So there's all different antigens on these MHC class 2s. So different antigens from the same pathogen might have activated these two different cells. So now the daughter cells for the B cell, some of them are going to be long-lived memory cells. You will, after this encounter, have an army of B cells that live for years and years in your body that can bind to this pathogen. Some of the daughter T cells will be long-lived memory cells that will live for years and years and years that can bind to this pathogen in your body. And so because of that, anytime you encounter this pathogen again, you don't have to go through this whole long process I just described. The pathogen will get in your body, it won't have time to proliferate, because instead of just waiting until it encounters the one B cell that could bind to it, there's going to be an army laying in wait for it forevermore. And so one cell gets in, it's attacked right away, it doesn't have time to cause disease in you. Same with the T cells. There's all these T cells so that as soon as a B cell binds to and phagocytizes this pathogen, the helper T cells, it's really fast now for the correct helper T cell to find those B cells, because again, you have a whole army of them laying in wait. So you get the memory cells that give you your immune system memory that are generated from this process, from this co-activation here. Another thing that happens is that some of these cells that are being produced, some of these daughter cells, are going to not be for memory, but for fighting the current infection at this very moment. So those are going to be the antibody generating factories for the B cells. So some of these daughter cells will not live a long time, but they'll make massive, massive quantities of antibodies and wear themselves to death really fast, just by spitting out these immunoglobulins. So remember, these antibodies are just the same immunoglobulin, but spit out into the body fluid. So instead of just sitting in the cell's surface, they're just going to be spat out as weapons. And remember, antibodies, there's 5 things antibodies can do to fight pathogens, even though they're not attached to a living cell, but they can neutralize, they can agglutinate, they can opsonize. There's cell-mediated cytotoxicity, antibody-dependent cell-mediated cytotoxicity, and what am I missing? Oh, activating the complement system. So there's all of these things that the antibodies just free-floating on their own can do. So some of these daughter cells will just spit out those chemical weapons to fight the current disease and die quickly, and then others will be the memory cells. These helper T cells, the daughters can be all different kinds of T cells. It doesn't necessarily, it's going to produce some daughter helper T cells, but other daughter T cells will be different kinds, maybe cytotoxic lymphocytes, maybe something else that we haven't covered. So you get a wide diversity of immune cells from the helper T cell proliferating. So if I were to try to draw that, oh, that's not right. Okay. So let's make this small now. Here's a small daughter. I'm just drawing the B cells small so that you can see we get an army of them. And some of them, again, are going to be long-lived memory cells. Oops. And some of them are going to be antibody-generating factories. And you can see this is getting a little messy now because this is not the best whiteboard program, but it's what I have for free. And then same idea with these T cells. So you're going to make a whole army of these, which I'll put along this edge here. All different kinds of T cells. Each different kind of T cell has a different job, produces different cytokines. Different cytokines do different things in the immune system. Some of them might dampen the immune response so it doesn't go overblown. Some of them will have other features. So, so that's what you have. You have enhancement of the humoral immune response here, and enhancement of, this is technically a piece of the cellular immune response, but it's designed to facilitate or to aid with the humoral immune response. And that's the end of that.